Hello, and welcome to the CMI webinar series designed for CMI and by CMI. I'm Cynthia Howell, and I'm the Outreach Training and Workforce Coordinator for CMI, and today I'm going to be your moderator. I want to tell you a little bit about this webinar and how we'll kind of conduct business. First of all, we are hosting all of these from Colorado School of Mines, and delighted to do it. And we also are always looking for new webinar topics because, again, this is for our CMI body. So if you have a new topic, please don't hesitate to put it on the exit survey. And also, this is a public webinar, so after the webinar is completed, it will be about a week, and it will be placed on the aimslab.gov/CMI under the workforce menu. This is a new place, so just keep that in mind. Now all of our webinars will be placed in the workforce menu. If you'd like to ask a question during the presentation, our presenter actually has asked if we keep questions in the Q&A area queued up so that she can address each one of them at the end of the presentation. So if you will abide by that, and do write your question as soon as you have it, because it does take a while to put it in that queue. And most of you remember that because you've been on these webinars quite a few times. I'd like to introduce our presenter today. Today, our presenter is Yongqin Jiao. She is a group leader of the Systems and Synthetic Biology Group and at the Biosciences and Biotechnology Division at Lawrence Livermore National Lab. She has her PhD from California Institute of Technology in Geobiology. She's a CMI principal investigator for the developing biotechnologies for the recovery of rare earth metals from low-grade feedstock. She's also been a PI on several DOE projects. She also has been an awardee of the Department of Energy Early Career Award, and that was in 2011. She has a lot of research interests. You're going to hear about some of those as she does her presentation, and I don't want to take any more of her time, so I'm going to introduce the presentation. Will be on the development of a biosorption process for the recovery of rare earth metals. And Dr. Jiao, if you'll take it away from here.
Thank you, Cynthia, for the nice introduction.
You're welcome.
Let me share the slides. Okay. So can everybody see the slides? Well, I will take that as yes. Thank you everyone for tuning in this hour. So I'll be talking about biomaterials and biotechnologies that develop for rare earth recovery and separation. And this is part of the project that belongs to FA 3.2.13 led by David Reed at Idaho National Lab. So first and foremost, I would like to acknowledge the team, including a young, energetic team that's shown here from Livermore. And I can promise that those photos were taken recently. And also this project will not be feasible without the very supportive and fun to work with collaborators from the various institutes shown in here. So here is the list of patents and relevant publications in case you are interested in reading about further. Here is the outline of the research topics that I'm going to discuss today, including the selective biosorption desorption process for rare earth recovery, and that includes from batch scale tests to cell immobilization PEA to enable a flow-through recovery in the column systems. And if time permits, and which I hope so, I'll introduce some work in progress that with the work that we conducted in the past 2 quarters, and then followed by summary and next steps. So in FA 3.2.13, together with Idaho National Lab, our goal of the project is to develop and integrate biological processes for rare earth leaching, recovery, and separation. And here in this graph, showing you a very simplified schematic diagram of the mass flow systems. That if we start with a rare earth source such as the electronic waste, so we leach the metals into solution through either abiotic acid leaching or bioleaching. And then once the metals are in solution, we apply a column separation approach for selective recovery and separation of the rare earths from the rest of the non-rare earth metals. And then we could subject this solution into a precipitation to form total rare earth oxides, followed by a rare earth separation step to produce individual rare earth elements. And for today, I'm going to first focus on the column separation for selective recovery, and later on touch upon a little bit about rare earth separation. So the biological ligands, or the secret source that we start with, are the two biological ligands that have previously found that bind rare earths with high affinity and specificity. And on the left, showing you the lanthanide binding tags that has been discovered over a decade ago by Barber and Imperiali's group at MIT. And here showing you that the rare earth is being coordinated by about 8 biological functional ligands supported by 6 amino acids in a very short peptides. Here we call it lanthanide binding tags. And this peptide we have worked with several years within the CMI work. And until recently, we started to introduce a new peptide called Modulin, which I'm going to touch upon at the end, towards the end of the presentation. So let's start with the lanthanide binding tags. So using the lanthanide binding tags, we have developed a selective absorption-desorption approach for rare earth recovery. And let me walk you through of this process. So through synthetic biology approach, we're able to engineer a microbe, a bacteria such as Caulobacter, and have the microbes to synthesize, transport, and display the rare earth binding ligands on the cell surface. And therefore, with the bioengineered cells, when we expose them into a solution mixture that contains rare earths and other non-rare earth metals. Because of the high affinity and selectivity of the ligands on the cell surface, the rare earths and only the rare earths bind. Upon binding, we separate the microbes from the rest of the solution that contains the non-rare earth metals using either centrifugation or filtration. And then with the recovered cells, we strip the rare earth off the cell surface using a diluted citrate buffer. And this step not only enable the recovery of rare earth, but also enable the recycle and reuse of the microbes for additional rounds of absorption, desorption. So one thing that I want to note is that the use of the microbes here are to synthesize and display the ligands, the rare earth binding ligands on the cell surface. And upon this step has been completed, the absorption, desorption, the rare earth recovery step is more like chemical process, and therefore cell viability is no longer important. So in this next slide, I'm going to quickly show you a couple of the performance with different feedstocks that we have tested. And in this slide is the acid Lychee solution from Bull Hill tailings from Wyoming. And the top graph showing you the metal composition of the leachate that we start with, and showing you that in the log scale on the y-axis is the metal concentration for the different metals in the x-axis scale, in the x-axis. So as you can see that it contains rare earths and a lot of the non-rare earth metals in higher concentrations. And at the bottom, the graph showing you the fraction of the metals that we were able to extract for different metals in the x-axis, showing you that we extracted and recovered most of the rare earths and removed most of the non-rare earths except a little contaminant of zinc. And here, this graph showing you the performance with a acid leachate of electronic waste, which is the from the speakerphone of mobile devices. In this graph, showing you y-axis is the metal composition concentration in micromolar, and x-axis is the different metals. As you can see that in the red columns is the lychee composition as a feed solution. And you can see that it has a lot of the non-rare earth metals such as iron, nickel, and zinc, and cobalt. And the blue column shows you after one round of absorption, desorption, the metal composition that in the recovered solution, showing you that we recovered most, if not all, of the rare earths, including PR, Dy, and Nd and Dy, and eliminate most of the non-rare earth metals. So far, the performance that I described are the batch scale solution where we separate upon biosorption, we separate the cells from the rest of solution using centrifugation filtration. And this step is energy intensive. And therefore, for the industrial scale application, it's important to develop a flow-through system, and in which cell immobilization is a key step. So for cell immobilization, we tested two approaches. On the left is through biofilm formation of having the microbes form biofilms on abiotic surfaces, such as the biofilm carrier discs. We call it MUTEC biochips. And the second approach we tested is on the right, is immobilize or encapsulate the microbes into the hydrogel beads. And this beads we call it microbeads. So very quickly, here shows the comparison of the performance for rare earth absorption using these two platforms. On the left is a crystal violet assay showing that we could form biofilms on those biofilm carrier discs. And on the right is a microbeads that with polymer packed polymer encapsulated microbes. It's a confocal image of the microbeads of about 100 micron in diameter. So as you can see from this table below, the rare earth absorption capacity in the per volume base for the microbeads is about 100 times higher than that of the biofilm discs. And therefore seems that microbeads is a more promising platform for rare earth absorption. And this notion is further confirmed by the technical economic analysis that's being performed by a collaborator from University of Arizona. So here, if you focus on the cost, the dollar cost per kilogram of total rare earth oxides that from these two platforms, you can see that the microbeads is far more profitable than the biofilm discs. And the biofilm discs has a higher cost because of two reasons. One is the low cell attachment efficiency during the biofilm formation, and the second is the low cell density of the resulting absorbent. So since the microbeads is a more promising platform, we developed further. And we developed further owing to the effort from a graduate student, Aaron Brewer from UW. He developed, so initially for the microbead formation, we adopted a fluidic method that previously we developed at Livermore. But then we quickly found that the throughput for of the bead production through the fluidic method was very slow. And therefore, Aaron developed a bulk emulsification approach for cell encapsulation to make those microbeads. And in comparison to the fluidic method that we used initially, this bulk emulsification method has increased the cell loading by 20-fold. That translates to rare earth absorption capacity directly, and has increased the throughput of production by over 10-fold. So the microbeads that we used could be used for resin beds for columns. And here, this slide is showing you a recap of what I described about our process so far. So we use synthetic biology to engineer microbes to have them display lanthanide binding ligands on the cell surface. And because the microbes are tiny in the micron size, they are hard to work with through a chemical engineering point of view. And therefore, we encapsulate them and make microbeads that's in the hundreds of micron scale, suitable for column study. So in this picture showing you a column that we poured with those microbeads as the resin bed. And you can imagine that when we have a solution with mixture of metals containing rare earths and non-rare earths, because of the selective absorption behavior of the microbes and the microbeads in the columns, the rare earths will bind to the column and the non-rare earths will flow through immediately. And that is by following the concentration of the various metals in the outlet. And that's we can make a breakthrough analysis. So this graph showing you the column breakthrough curve with the feed solution is a mock solution that contains neodymium only. So on the y-axis of the breakthrough curve is the concentration of a metal versus the ratio concentration of metal versus the initial concentration in the feed solution, and one is 100%. So as you can see that with the microbead poured column, the rare earth is being captured in the column until the absorption capacity has been reached and the rare earth starts to break through. And in contrast, if we have a column that has only the polymer beads without the microbes, which is the no cell bead control, as you can see that there's no absorption capacity and the rare earth started to break through right away, suggesting that indeed it is the microbes in the beads who are doing the absorption. And the graph on the right showing you that after absorption of the column capacity has been reached, we use a diluted citrate solution to elute the rare earth from the column to recover the rare earth. And you can see it's a sharp peak of the neodymium came out rapidly. And in comparison, this breakthrough column is, this breakthrough curve on the left is replotted here in black, showing you that this desorption step not only recover 100% of the rare earths, but also we were able to concentrate the rare earths, by several fold. And here, besides the mock solutions, we also tested the electronic waste leachate. So again, a breakthrough curve showing you on the left. So this inset showing you the major metals present in the electronic waste leachate that we use as a feed solution. And again, the breakthrough curve on the y-axis is the various metal composition or concentration that break through over time in the outlet over time that we converted to bed volumes. So a couple of observations from this breakthrough curve that I like to note. One is that the breakthrough time of the rare earth is very different, is much later than that of the non-rare earth metals, indicating selective recovery. And second is that right at around the time of rare earth breakthrough, you can see that the concentration of some of the non-rare earth metals has a second peak here, such as the nickel and zinc. And this, we think, is likely caused by competitive displacement of the non-rare earth by the rare earth when the column is become saturated with the rare earth. And the third observation is that the breakthrough time of the rare earth metals all occur similar time, although with different slope, indicating that this absorption-based approach is not likely to be an effective method for rare earth separation. So the graph on the right is a replot of the data from the left, showing you that if we were to stop the column at any time point of the breakthrough curve, what is the content or the product or the metals that's being trapped in the column? So here on the blue, showing you the fraction of the rare earths is being trapped in the column, and showing you that it's almost 100% being trapped or recovered until the rare earths start to break through. And the red curve showing you that the purity of the product, which is the total rare earths compares to the total metals present. And it starts to increase as the non-rare earths start to break through, and a second increase, a slower slope here, could be likely caused by the competitive displacement that I mentioned earlier. And right around the breakthrough, the rare earth breakthrough time points, we were able to achieve over 94% of product purity and have very high, close to 100% of extraction efficiency. Right, so in the next, the remaining time, I'm going to update you with some of the very recent work in the past 2 quarters, our effort towards rare earth separation. So if you were like us, brave enough, we're thinking with this column, what happens if we put a rare earth solution that contains 15 lanthanides at equal molar concentrations? So this is the breakthrough curve look like. As you can see, again, consistent with the electronic waste leachate, all of the rare earths breakthrough points are very similar. And this inward, you can see that this inward, towards this direction of my mouse curves, that shows the high is from the lower affinity of the lanthanide binding tags to rare earths towards higher affinity. And this slope is consistent of what we observed in terms of lanthanide binding tag rare earth complexation affinity that we previously observed in solution phase. And again, also if we see that the weakest rare earth lanthanide binding tag complexation, which is the lanthanum here, has a second bump here, indicating again a competitive displacement of the lanthanum by the heavier rare earth that over time. So overall, again, this experiment shows that purely absorption-based approach with our column is not likely to give us a good separation for the individual rare earths. And therefore, we turned into a desorption-based approach. So we checked what about sequential desorption for rare earth separation? And I'd like to note that this experiment showing here is batch analysis, not in the column, which means with free-floating microbes. So with the lanthanide binding tag engineered on the cell surface with the of the bioengineered microbes. So in this experiment, we first immobilized or absorbed 15 of the lanthanides at equal molar concentration on the cell surface. And then we subject the rare earth bound cells to solutions that contain a desorbent, which on the left is 2.5 millimolar of oxalate, and on the right showing you with the desorbent being 200 millimolar of bicarbonate. By the way, so those concentrations are the best ones that give us a better separation plots that I'm showing here. So as the y-axis in those plots are the fraction extracted for individual rare earths, and axis is the atomic radii of the different rare lanthanide series. So you can see that with the oxalate, we're pretty much more or less show a linear relationship from the heavy to the light rare earths. And in the contrast, with the bicarbonate, we saw a steeper slope from the heavy to the mid rare earths, gadolinium or europium. And then there's not much a pretty flat line from mid to light rare earths. So these patterns, we hope to provide a basis for selecting of exact and optimizing of conditions, the desorption conditions, to achieve better separation for feedstocks with a specific rare earth composition. So, and also another note I would like to mention is that in this experiment, for any metals or any rare earths, whether they stay in the stationary phase, which is on the cell surface, or they decide to come off the surface and join complex with the ligands in the mobile phase, which is oxalate here. And that decision is based on the relative affinity of any individual rare earth to the stationary versus the solid versus the mobile phase. And the rare earth complexation with oxalate or bicarbonate is well known. And in contrast, the rare earth complexation with microbial surfaces is much less known. And to further understand the mechanism of rare earth complexation to bacterial surfaces, our collaborator UC Berkeley has helped set up a cell-surface complexation model to describe the rare earth binding behavior on cell surfaces. With on the left showing you the reactions that they have considered for the complexation. And as you can see that so I'd like to note that for bacterial cell surface, besides the lanthanide binding tag that we engineered on the cell surface, there are a lot of native functional groups on the native bacterial surface also contribute to a significant fraction of the binding. And for example, in this chart showing here, some of the carboxyl, phosphyl, and hydroxyl groups that are present in different concentrations and with different pKa's, and those also contribute for rare earth absorption. And as you can imagine that if we were to use this system for rare earth separation, we have a very complex ligand system to work with. And to that end, a chemical engineer postdoc, Jesse Dong, recently joined us. He thought about how about let's forego the bacterial surface and have a clean surface that we just directly use the lanthanide binding tag and construct the immobilize those tag on the abiotic surface and have a cleaner surface to work with for the separation purpose. So this is exactly what Jesse did. He chose two different platforms of the agarose or the MCF, which is mesocellular foams, using chemically immobilize the lanthanide binding tag on these two surfaces. And he compared preliminary results, shows that the rare earth absorption, sorry, I have to do it else here. The rare earth absorption with the different, with these two functionalized, the lanthanide binding tag functionalized surfaces, the MCF and the agarose beads. Preliminary results already show that, is already showing that the rare earth absorption capacity is 2 or 3 times higher than what we're able to achieve with the cell immobilized packed microbeads that I previously mentioned. So this is really promising, and Jesse is going to present this work at the CMI winter meeting for more details, with a lot more details and data. So far we have focused on the lanthanide binding tags, and another ligand that we recently added to the toolbox is called Modulin, abbreviated LAN-M. So this is in collaboration with collaborator at Penn State University. So Modulin is originated as a rare earth binding protein that originated from a small protein of an unknown function from methylotrophic bacteria, and this group of bacteria has been discovered living in the volcanic ash environment, and is the first group of bacteria that's known so far to use lanthanides as a for biological functions. So there are a lot of reasons why we want to add Modulin to the toolbox, and I'm going to mention one of them today. That is Modulin binds rare earths with super high affinity. And this work is pioneered by a postdoc, Gautier de Blondy, who is also going to present at the CMI winter meeting. So in this plot, showing you showing you the LAM binds rare earths with high affinity. With the y-axis is the Kd dissociation constant in the unit of nanomolar, and the y-axis is ionic radius to differentiate the different rare earths. So so the dissociation constant is inverse relationship with the log beta, which is the affinity, meaning that the lower the number, the higher the binding affinity is. So right away, you can see that the Modulin rare earth binding affinities are several order of magnitude higher than that of the lanthanide binding type rare earth complexation. And also, you can see that the binding affinity across the rare earths shows different patterns with these two ligands, indicating that those could be explored for rare earth separation. So in summary, I described a selective biosorption desorption process that could be used for rare earth recovery that we have tested with different feedstocks. And we have, using the encapsulated microbeads, we poured column and showed promising results from different feedstocks using the column. And also we have tested a few of different using sequential desorption for rare earth separation that's been currently underway. And we added Modulin in our toolbox because of high affinity and rare earth binding affinity, among other properties. So the next steps, we're going to continue to focus on chromatography development for rare earth separation, in which we have two main approaches. One is to adapt the microbeads column that we have set up for sequential desorption, changing the desorbent concentrations and compositions, and testing with real feedstocks. And the second is to adding the Modulin, and along with previous work with LBT, immobilize on the abiotic surfaces, and using this immobilized platform for rare earth separation. And because we have worked with the LBT for several years, and for Modulin, there are more characterizations of background work is needed, and that is summarized that's currently underway and summarized in the second bullet point here. Such we're going to continue to characterize the Modulin rare earth complexation in collaboration with PSU on the protein engineering, where they help shorten the protein for more economic protein synthesis, and doing site-directed mutagenesis to enhance the selectivity among the rare earths. And then we're doing some characterization and testing with real feedstocks. And thank you everyone for listening, and I welcome any questions.
Yongqin, thank you so much for the great presentation on development of bioabsorption process for the recovery of rare earth metals. You presented a lot of promising processes, including your work on the microbead for just being, again, a promising platform. Thank you for the work update into the future as well. That's going to stoke a lot of questions here. And it sounds like for the winter meeting, we're going to hear more about this promising future research. So it does look like we have quite a few questions. If you'd like to start with the first one from Mark.
Yep. So Mark had asked, have any companies expressed interest in scaling up this technology? So the answer is yes. So there are, so we have been in contact with some companies, and one recent ones is in Solvay. So for example, in making the microbeads, although in the laboratory scale, that's pretty fast. However, in industry scale, they may or may not be the exact platform that they're going to adopt for the cell encapsulation. And for that, we've been in conversation with Solvay to see and to further develop an industrial relevant process that's more economical for cell encapsulation. So, but with that said, we are still going and trying hard to find more industrial partners to develop the project, so that, and also make sure that we are working on the right track towards industrial applications. So if anybody have leads of biotech companies, chemical engineering companies, feedstock companies that could be helpful for our project, we welcome any of your input. And second question from Mark, do you believe that 30% discount from the rare earth price to be a reasonable assumption for a TEA for a mixed rare earth oxides? So I'll answer the question first. Maybe Hongyue from UA, who helped with the TEA, could answer the question better. So I believe that this practice, whether it's reasonable or not, is up to debate. And I think she adopted this based on the references from other studies. So Hongyue, could you please pitch in on that? Right, so maybe Hongyue is not, sorry, Hongyue maybe is not on the line, but yeah, so may or may not be reasonable, but we follow the previous studies. And David really commented on nice presentation. Thank you. Thank you for your support and collaboration. And David has another question. Can you clean the column for indefinite usage? So the answer is probably no, not for indefinite usage. And but the column usage is one of the most important parameter for this technology, for the economic prospect of the technology. So here on this graph, showing you that with the packed microbeads column, we tried, or Aaron tried 9 times of absorption, desorption, and the same column, absorption, desorption again. So he tried 9 cycles, and this is what the breakthrough column looked like. So we found that about 85% of the absorption capacity was remained, and in this packed column. So and I think the performance about level off. So this is another thing that we need to add to the TEA about the sensitivity analysis of the reuse, recycle, the ability of the reuse recycles, and how these factors play into the TEA. And we expect this column should be used by hundreds of times, and that needs to be demonstrated. And David has another question. Can you comment on the disadvantage for using the LBT and a Modulin sequentially or together? So as we can see from the dissociation constant or the binding affinity analysis, that Modulin are 3 or 4 times orders of magnitudes higher affinity for the rare earth. So if we were to use the 2, biological ligands together, we could we could think about especially for separation purpose, that we have one as a mobile phase and the other as a stationary phase, and play around with their different rare earth affinity patterns among the lanthanide series, and to work towards separation. So that's one scenario could be explored. And together and sequentially, I have not thought about using them sequentially yet, but David, we can talk more about how to work with to set up those different scenarios. Okay, so moving on to Yoshiko's question. Let me read it. Have you done tests to show that the abiotic immobilized LBT also have advantage of higher selectivity for rare earths, since there are not the other non-respective binding sites that occur on the cell surface? So I think that for this question, I mean, Jesse can pitch in that we have not exposed the LBT immobilized on the abiotic surface for complex feedstock yet. However, we have tested the Modulin with a electronic waste leachate. So the reason for that is the immobilization work is only happening recently, and the Modulin is such a tiny peptide. So if not, without immobilization, we will not be able to, well, easily separate the peptide from the rest of solution after absorption. And therefore the LBT type of work can only be enabled with a solid developed immobilization system. However, this is not the case for Modulin, because the Modulin relatively is a larger protein, and therefore after the binding, we could use a spin filter to separate the Modulin from the rest of the solution. And long story short, that we tested Modulin in the solution phase with electronic waste leachate, and we do find much better selectivity towards rare earth, total rare earths, not the separation yet, and comparing to the non-rare earth metals such as iron and zinc that we show in the, so Gautier is going to describe all of that in the winter meeting. There you can find a lot more detail and exciting results with Modulin. So next question is from Dennis. Thank you, Lychee, for the nice work. Thank you so much for the comment. My question is, what is the capacity in terms of gram of bees per separation column? 100 times 1 centimeter. The capacity of the bees, so it varies based on the feedstock and based on, so I think slightly based on the condition we tested, but I would say that it's about 10 to 15 milligrams per gram of absorbent that we normally see. And during manuscript review, submission and publication, so we got some comments from the reviewers about the relatively low absorption capacity comparing to other absorbents such as the granulated activity carbon, the gag column. And then our response to that would be that, yes, the other non-selective gag columns have a lot higher absorption capacity. However, what they lose or they pay the price is that they have very low selectivity. So the selling point of our columns of our absorbent is on the high selectivity part. And yes, so I agree that the absorption capacity would be could be improved. Okay, so another question from Yoshiko. Which specific lanthanides are most often found in mesotrophic lemodulins? That's interesting question. So some of the rare earth eating methanotropes that they found, I think in the literature was reported is that they were only to utilize the lighter rare earths. A few of them are from the lanthanum to a few of the lighter ones for reasons that's unknown. And however, when we tease out the biological native environment and the whole microbes, there are a few of the proteins involved in that pathway of microbes eating the metabolizing the lighter rare earth, and these proteins surprisingly have different absorption, different complexation affinity with all of the rare earths. So the conundrum is that those microbes can only survive on the lighter rare earths. However, the chemistry works for all of the lanthanides. So it is problematic for microbes. However, if we extract the chemistry and utilizing them for the biotechnology rare earth extraction separation scheme, it doesn't seem to be a problem. So one thing I want to add is that besides limodulin, there are other siderophore-like proteins, siderophore-like small molecules that's also been produced by the same group of metallotrophic bacteria that other groups are looking at for biotechnology application of rare earth extraction as well. Question from Bruce. You're making good progress. Thank you, Bruce. Can your separations be accomplished from acid solutions? So I'm glad you asked that. I would say depending on how acidic solutions that you refer to, and because of proprietary information reasons, I think I can defer answering that question maybe during the CMI meeting, and also Gautier, because it's a more closed meeting for CMI, Gautier is going to include a lot more of the information about the conditions that limodulin would work. Okay, so we have another question from Homia Yang here. Excellent work, thank you. And can you explain how this technology is different than bioflotation? So first of all, I have to admit, I don't exactly know what bioflotation is. But if I have to guess, would be the biomolecules such as the biosurfactants that has a carbohydrates are the sugar module that could complex with metals and followed by a long lipid tail. So those biosurfactants, based on the literature, they various them has been discovered could be produced by a lot of different microbes, especially environmental ones, and has been shown for bioremediation of heavy metals such as copper and zinc, etc., especially from the soil environment. So in this regard, those biosurfactants that use similar to other surfactants used in flotation, the module, the carbohydrate module that binds to the metals, at least in the biosurfactant case, are less are carbohydrate based instead of protein based. And from my literature reading is that those are less specific. They bind metal less specifically than the two biological peptides or protein ligands that we're looking at. And also it's a biosurfactant, and similarly to the other surfactant utilization, they in the extraction, if we were to apply to an extraction scheme, you would use a lot of the solvents, and then for the binding and flotation separation to occur. So it's another, I would say, the biosurfactant case is for bioflotation is another pathway of using biological ligands for rare earth, for metal recovery or bioremediation, and is in parallel with what we're working on for the absorption desorption approach. And thank you for all of the questions.
Yongqin, thank you so much again for a great presentation and the promise of more of that research findings in our winter meeting. That's going to be great for all of us. We'd like to thank our participants for attending, and again, exceptional questions that kept you quite busy at the end here.
Yep.
I'd like to remind everybody, if you have a suggestion for a future webinar, please include that on the exit form with your contact information. Our next CMI webinar will be on Wednesday, February 26th. Bruce Moyer from Oak Ridge National Laboratory is going to be presenting on the role of research and technology maturation, perspectives from a researcher, technology developer, and research portfolio manager. And of course, as always, details are going to be in the critical times and at aimslab.gov/CMI. Thank you again, and we look forward to seeing everybody in our next webinar on February 26th. Thanks so much.
Thank you, Cynthia.